Our research. Early screening of hemolytic risk in functional peptides via Ensemble AI using sequence and concentration features. Therapeutic peptides or antimicrobial peptides with high hemolysis risk are unsuitable as drugs, but experimental validation is costly. Our model enables early in silico screening to filter out hemolytic candidates. We designed an ensemble model with advanced data preprocessing, integrating sequence and concentration inputs using two encoding methods, PC6 with CNN and PepBERT with MLP. Our model outperformed existing AMP predictors in two test set evaluations. Furthermore, it accurately captured increased hemolysis risk for identical peptides at higher concentrations. Dataset. The data was obtained from VASP database, containing peptide sequences, concentrations, and hemolysis percentage. A clinical threshold of 10% hemolysis was defined to label samples as hemolytic, also referred to as positive. After collecting sequences containing 10 to 50 residues and removing samples with non-natural amino acids, we obtained 2,894 samples. They will undergo similarity-based preprocessing in next page. We also prepared an independent test set, called Test Set 2, consisting of 18 peptides designed at 7 different concentrations, with hemolysis validated using sheep blood in our wet lab. Data preprocessing. Considering the challenges of peptide synthesis, we set the maximum peptide length to 32 residues. For similarity-based filtering, CD-HIT 2D mode was used to remove redundant sequences with over 90% similarity across 4 concentration clusters. CD-HIT 1D mode was then applied to select representative samples within each cluster, yielding 935 positive and 995 negative samples. In first AI model, it utilized PC6 encoding and a CNN, incorporating not only sequence and normalized concentration features but also their element-wise multiplication interaction to enhance joint representation learning. Concentration values were normalized using min-max scaling over the range 0 to 240 m. And PC6 values were derived through z-score normalization and min-max scaling. Finally, the array has 13-dimensional features input as shown in right figure. The second AI model utilized PepBERT small encoding with normalized concentration as an added feature. That's total 161 features. Diverse key information was captured using 3 pooling types: global average pooling, global max pooling, and global minimum pooling, followed by batch normalization to reduce excessive value variation. The resulting output was fed into a multi-layer perceptron. Soft voting with a 0.5 threshold was used for model ensembling. Samples with output probabilities above 0.5 were classified as hemolytic. Comparison with the current study. In Test Set 1, 10% dataset, the model showed balanced performance on both positive and negative samples and outperformed existing AMP predictors. In Test Set 2, our wet lab validated data, it also outperformed existing tools and accurately predicted elevated hemolysis risk for the same peptides as concentration increased, as shown in right figure. To align with the positive sample definitions used by other predictors, we adjusted the sample labels in both test sets accordingly. For comparison purposes, a concentration of 50 micromolar, a typical clinical dose, was assumed when necessary. Label definitions for each tool are detailed in the supplementary QR code in next page.